So yesterday we found out that cells can divide, and we can get new skin cells from old skin cells, and we need a certain type of cell to make a new cell. Today we want to look at what do they need to be able to do that, and where do they get that stuff, whatever it is. So we had a quick discussion. We found out that we probably need food or energy to have these cells do this stuff. And like our bodies, they would probably get it from the food we eat. So somehow the energy and the food that we get, somehow it gets to the cells. We wanted to see if this is true, so we can kind of check out, we could maybe take a cell from our body and see if it needs food. But the only problem is if we take it out of our body, then it would die because it's not part of our body anymore. And I don't want to test it on your body because I'm hurting you, because I'm killing part of your cells off. So one idea we could use is find an organism that's only made of one cell. And one of those things we could look at is a bacteria. And so we kind of talked about what we know about bacteria, that some are good, some are bad, they're all over the place, they can cause disease. So we just had a couple of things about that. We said, maybe if you get too much bacteria, it might cause an infection and a wound. So we looked at those things. To take a look at some images, you can see that we have some bacteria on the left-hand sides. So they're different shapes. These are kind of round. These are kind of like lines. They're definitely smaller than cells that we have on the right-hand side. So different shape, different size. Don't worry about the color, because remember, we might sometimes stain cells so we can actually see them. So just to compare a bacteria versus a cell. But now we want to figure out, how do we get evidence to show that bacteria really needs food? So the general idea that we came up with in all classes was, well, let's take some bacteria, let's give it food, and let's give some bacteria no food. If it really is using the food, the ones that don't get any will die. And if it is getting food, they'll grow. So it kind of makes sense. So we're going to have to figure out how we do this in a scientific way. So scientists use E. coli bacteria, very easy to grow in a lab. They usually don't cause disease, so we don't have to worry about a disease getting out of a laboratory. And what happens with E. coli usually happens with a lot of other cells. So wherever we find out happens here, we can kind of learn a lot of other things. To do this in a lab, we need to make agar, and agar is a dry powder made from red algae. So you take red algae from the ocean, you dry it out, you grind it up into a powder. You take that powder, you mix it with boiling water, it dissolves, and when it cools off, it kind of forms like a semi-solid jelly, and then they put bacteria on that agar and let it grow. So let's watch a quick video about how we make this agar stuff. So this is how we make agar plates. So in the vial is some hot agar, and here's our petri dish where we're going to put the bacteria. So just kind of mixing up a little bit there. He's putting a glove on not because it's dangerous, it's just that the agar is still hot, so it's liquid versus that semi-solid jelly. It's pouring a little bit in. Not trying to fill it, just covering the bottom, and he's going to stop. He's going to put the lid on of this petri dish. Going to let it cool off, and you'll see how it doesn't flow or anything, it's just there. In our next video, we get to see what this looks like. So he's going to kind of take this little spatula and peel the agar out of the container, so you can kind of see what it looks like when it's not in the dish. So he's just kind of scraping it off the bottom there. And you're going to find it looks like a piece of thin rubber. So you can peel it right out. You can kind of flex it. I guess if you made a really thin layer of Jell-O, it might look like this. So now we want to show how bacteria might actually use food. So we take this agar, we mix it with some nutrients, some food. So they set it up so the first tray had 1.15 grams of just plain agar, no food in it. And then they made another one. If you add up 0.29 plus 0.86, you get 1.15. So it's the same amount of stuff going in the petri dish but we're just changing what's going into the mixture. So what we do is we keep changing this. And I'm going to show you what the percent nutrient agar on a sheet of paper here. So if we take the same numbers we just saw in the graph, if you take 0 grams of food divided by 1.15 grams total of the stuff that's in the Petri dish, you get 0% nutrients. If you take 0.29 divided by 1.15 by the total, it's 25% food. 0.58 grams divided by 1.15 grams gives you 50% food. 0.86 grams of nutrients of food divided by 1.15 grams of stuff gives you 75% food. And then finally, 1.15 grams out of 1.15 grams gives you 100% food. So we do this and we check to see what happens with the bacteria. So now that's how they made the stuff. Now we got to put some bacteria on it. So let me watch another video here. So what they're going to do is take this tool that has like a little loop on the end, and they're going to put some bacteria on it. So I'm going to kind of walk you through. So there is the tool with a loop. They heat it over a flame so it kills any other bacteria or anything that's on the loop right now before they even started. So that's killing everything. They bring it back down here. They're going to let it cool off a little bit. It's going to touch it with the agar, just cooling it off so we can actually do something with it. This is the E. coli in this test tube. So he's dipping the tool into the bacteria. And he's going to rub it on the top part above that blue line. That's where the bacteria is. He's going to sterilize it again, so he's killing everything that's on there. He's going to cool it off again on the side. So there's no bacteria on it, he just killed it all. And what he's going to do is he's going to make a line down the middle. We can't see it, but he just made a little line going down the middle. He's going to clean it off again, sterilizing it, heating it up so it kills everything. And what he's going to do is kind of go back and forth really quick, just making like a zigzag on the agar. So he's just cooling it off there, and then here's a zigzag. Okay. And he's going to cover it up, and we're all set. He puts it into an incubator, which is at 98.6 degrees, our body temperature. So whatever happens here is something that could possibly happen in our body because we're the same temperature as that. It's going to leave it in here for 2 days, and we look at our results. Now, normally we would kind of predict with no food it should die. With more food, we should see more growth. So here's our results. With no nutrients, I don't see any bacteria. With a little bit of food, we see some white blotches up here and some blotches down here. That's the groups of bacteria forming, so we can actually see them. Not one bacteria, just there's thousands in there. When more food, we're getting more bacteria. And this is where he kind of did that line down the middle in the zigzag. It just gave them some place to kind of start growing. When we add 75% nutrients, even more growing, and 100% nutrients, a lot more growth. Now I'm going to switch over to kind of turn this into a graph of sorts. So if you look here, when we have no food, there was no bacteria. When we added more food, we kept getting more growth on it. And WIS kind of stands for what I see. So we're going to just interpret this. So I see that the more food I add, the more bacteria that I get. That's what I see. What I mean, W-I-M, or sorry, what it means is that as we give bacteria more food, it allows them to grow more. And if we take away food, we get less growth. So what I see is what does the graph show me? And then what it means is what information do I get out of the graph when I start thinking about what it's about? So now we got to find out, does this have anything to do with any other single-celled organisms? So your last task is just to read an article. You get to pick one of 4, and you're just going to do some highlighting. I have 3 questions I want you to highlight some information for. So let's take a look at your options. So you're going to go to Schoology. So in Schoology, you're on the Day 16 page. You see the 3 videos that we just watched. And then there is attachments. So there's 4 you can pick from, and you only have to do 1. So anything that has the word reading at the beginning is your options. 1 is about algae growing in a fish tank, which is a single-celled organism. You can talk about yeast, which is what we use in bread to make it. Yeast is a single-celled organism. You can read more about bacteria, and then you could read about bacteria that live in very extreme conditions, like at the bottom of the ocean or near volcanoes. So what you want to do is pick one of those, download it, send it to Notability, and then highlight information that would answer one of these questions. So what did you see about the thing you read, the yeast or the Archaebacteria, whatever it is? What did you find that was similar to or different from, the skin cells, the muscle cells, the bone cells, the E. coli? So some similarities and differences. If you can also find some information about how your organism's structure and function helped it live, highlight something there. And then if there's anything that could help us talk about the healing process in people, maybe a little piece of information that would help us put a puzzle piece together, highlight that. And then we're going to talk about those in the next lesson.